so this is the second video of today's session. So as I said, we're going to search for a gene, and the gene we're going to search for is MCM6. So I'm just going to put it in the search box, and I'm going to go straight to the gene tab. So in the session last week, we talked about looking at sequences. So if I go to the sequence page, one of the things that we can see is the sequence of this gene with the variants plotted onto it. So we've got our sequence with our variants, and if I wanted to find out more about one of the variants I've found here, I could click on it. So you'll notice where we have the variants highlighted in the sequence, they're not A's, T's and G's, they're actually written as IUPAC ambiguity codes, these R's and S's and things like that. I never remember what they stand for, but if I put my mouse over them, so my mouse is currently over this S, and it tells me that S indicates C/G. Now I'm just going to click on this, which will get me a pop-up, which will take me to more information about this variant. I can click here. I've already got things like location. One thing to point out is I've now got different alleles to what I saw in the hover over. So it's now giving me my alleles as G/C, whereas in my hover over, oops, there we go, it's C/G. Now the reason for this switch is actually this strand here. We're on the reverse strand. So in the context of this sequence, in the gene sequence, the relevant alleles are the reverse strand alleles. However, this pop-up is taking us to the database entry for this variant, and the database entry always gives us the forward strand. It is actually highlighted there as well, so you know that's what you've found. So we can view all the variants as a table as well. So over here in the menu we have variant table. And this table lists all of the variants which are found within this particular gene. So we can see the variant identifier, the location, the alleles, the minor allele frequency if it's known. These evidence icons are really useful. Some variants will only have a single submitter, there's not a huge amount, and they might not of data behind them, they might not be real. So you can use these to exclude those, or you can just say, aha, this one's got data from Nomad, so this will have things like allele frequencies and things like that. We also have our amino acid change for these missense variants, and our SIFT and polyphen scores. And you'll see that they're highlighted in the red and green to indicate whether they're dangerous or not. If you are red-green colourblind, you can just put your mouse over them, and you'll get this little pop-up saying, so this one is deleterious. So if you're not sure, please do take a look. And you'll see that SIFT and polyphen don't always agree with each other. At the moment, we've got a filter on our table, so it's actually filtering by missense consequences. So if I open this up, you'll see that I have a list of all the different consequence types, and missense is currently turned on. If I turn on, say, splice acceptor variant and hit apply, I'll now get more variants appearing in my table. Annoyingly, not anywhere near the top. I can also filter by things like SIFT and polyphen, minor allele frequency, or other columns such as the evidence status, amino acid coordinates. So this can be a good way to find a variant if you've found a reference to it and you want to find it easily, and it says it's in this gene and it affects amino acid, whatever, then this can be a quick way to find it. So you can also get structural variants if you're interested in larger scale variants. There is structural variants in the table here. If I click here, you'll see that we have a graphical display showing these larger scale variants, and also a table listing them with a description of the study in which they were found. There's one thing I want to show you which is actually in the transcript tab. So I'm going to open the transcript table and go to this first transcript here, and I'm going to go to this haplotypes view. So this haplotypes page is taking data from the 1,000 genomes, and it's calculating the frequencies of the actual proteins which are found in individuals. So it's not just looking at variants on a singular level, it's saying how frequently do we see the combination of this variant, and sometimes even a third variant in this protein and what's the actual protein sequence that we can see. So if we scroll down, we have the reference, and we can see that in this case, the reference is seen 98% of the time. But there are some cases where the reference is not very frequent. We then have some of the haplotypes. So we've got some single variant haplotypes. We've also got a haplotype here, which has got 2 different variants. So it's saying that in 2 individuals in the whole of the 1,000 genomes, both of which were African, we see both of these variants occurring in one individual. So let's open up that one. You see that one of them is from the African ancestry in southwest US, and one of them is from the Gambia. And we can see in the sequence, there's one of them, and there's the other one. We can get the new resultant protein sequence, and we can also see which samples in 1,000 genomes these are. So if I wanted to order these cell lines from Coriell, for example, of these individuals to do some analysis on this particular protein, I could do. So now I'm going to go and explore the location tab. And just point out some of the things I showed last week. We have our variation data shown at the bottom of this page. The legend for the variants is shown here. And if I open up the Configure This Page menu and I go into variation, you can see that I can turn on variants from different sources. So I can turn on just the exact variants or decipher variants, so these are linked to rare disease. Or I can turn on 1,000 genomes variants by superpopulation. I can turn on variants associated with disease. I can turn on genotyping arrays. I can view those as well. And then again, I've got structural variants, which again, I can do by different study as well. So what I'm going to do, I'm just going to leave this, and I'm actually going to search for a particular variant that I'm interested in. The variant I want to look at is RS4988235. It's actually found in MCM6, but it's easier to find it this way. If I know the identifier, always search for the identifier. So this takes me to the variation tab, and you'll see I've got a menu down the left. I've also got these pretty buttons here, which actually take me to the same place as these menu on the left. I've got a lot of information anyway. I've got things like the minor allele frequency, my evidence status and things, but I'm going to explore some of these links. So the first one I'm going to go to is genes and regulation. And this shows me that it is in fact found in MCM6, and it's an intron variant in various different transcripts of MCM6. It's just loading the gene expression correlation, but we know it's not found in any ensemble regulatory features or motifs. And you can see that it has effect on a number of different genes. It changes their gene expression level. It has quite a large effect on this variant in the aorta and in whole blood, for example. So I'm going to use this menu on the left to explore, and the next thing I'm going to go to is population genetics. And it turns out this variant is very skewed across populations. So I'm afraid that we've encountered a bug here that we do know about, and we're finding really, really difficult to squash. These should actually be pie charts indicating the allele frequencies across different populations. We do have the data replicated in these charts here. So we've still got the data available to us. So this variant is very skewed across populations. The A allele is uncommon overall, and it's particularly uncommon in the African and the East. It's not seen at all in the East Asian population. What's quite interesting is in the European population, the A allele is actually very, very common, which makes it very different to the other populations out there. So this is the 1,000 genomes, and you'll see the 1,000 genomes data. We have allele frequency, and we also have genotype frequency as well from 1,000 genomes. If I scroll down, you'll see we also have nomad frequency. So we have much larger populations. We only have superpopulations from nomad. We don't have these subpopulations. And we also don't have the genotype frequencies from nomad. We only have the allele frequencies. There are other data as well as TopMed, UK10K and HapMap. But the most comprehensive is probably the 1,000 genomes here. So we know that this is a very skewed allele frequency. If I go to phenotype data, we'll see that this is actually a very skewed phenotype as well. So we can see that this variant is associated with lactase persistence, with the A allele, which was the one that was largely found in Europeans but completely absent from East Asians, being associated with lactase persistence. There's also links to body mass index and hip circumference, which I can only assume is due to the amount of cheese that people eat who can eat it. And it points out that it's found in the gene MCM6. If I click on lactase persistence, I can actually see everywhere in the genome that this phenotype has been associated. It's currently filtering by gene, but if I get rid of that, I can see all the variants that it's been associated with. The last thing to point out is the phylogenetic context, which is where we get ancestral alleles from. So if I click on this in the menu, we get an alignment of the region. Our variant of interest is highlighted in green, and we can see that all our primate relatives also have a G at this locus, so they are also lactose intolerant. So that's the end of this demo. You've now got an exercise to work on looking at variants in Ensemble, and then we'll come back and we'll have a look at the variant effect predictor.